Okay, so chapter 6 is about the viruses. It's an introduction. A lot of details, but not everything. We will have some more details in later chapters. So, generally speaking, this is just an introduction. The virus is an obligate intracellular parasite that's composed of two components, basically. An inner part and an outer part. The inner part is the genetic material, and the outer part is a protein coat. Some viruses will have additional envelope, and some others will not have it. But what obligate intracellular means?
Like inside the cell?
They
has to survive inside the cell?
Exactly. They have to be inside the cells. You leave it alone. If it's outside of the cells, they will not be able to do anything. Which order or family does this virus belong to? Is it bacteria, archaea, eukarya? Is it to any one of those?
No.
None. It's none of those. Okay? Because basically, the virus is not even a cell. In order to say something is a cell, you have to have two components, right? In the center, you have to have nucleus, and outside you have to have a membrane. A membrane. Wall can be an additional thing, but you have to have an official nucleus, a regular nucleus, genetic material, and a nuclear membrane, right? And you have to have a cell membrane. If you don't say that, it's not a cell in the first place. So this is not a cell, right? It's just a particle that's obligate intracellular, and the two components are genetic material in the middle and surrounded by a protein coat. Some of them will have an envelope and some will not have an envelope. I did not say membrane, right? I'm saying envelope, which is different. So viruses are obligate intracellular parasite, like I said, and the outside protein is important. The importance of that protein that's outside is to attach the virus to the host cell. Obviously, obligate intracellular, they have to find some cell to live in. In order to recognize and attach that cell to that cell, you have to have that protein which anchor them, attach them. And then the virus is going to get into that cell somehow. We will discuss the details. And then it's going to replicate itself. And we will discuss all these details. So if you look at this picture right here, this is the virus. What do we have in the middle here? Genetic material, and what's outside? Protein coat. Some of them have envelope and some does not have envelope, right? So this is how they attach in the first place. So they will get into the cells. They are going to replicate to create more viruses, and these viruses are going to leave the cells. So the virus go to the cell. The protein will anchor them into the cells, right? They will enter inside the cells and then replicate. What replicate means? Reproduce itself. Yes, make more copies of itself. Okay? And it's going to use the cell as a tool to reproduce itself. Okay, if you guys remember when we talked about the bacteria, and is bacteria prokaryote or eukaryote? It's a prokaryote, right? Why? It has a cell membrane, but does it have a nuclear membrane? No, it just have a genetic material, right? And this is the clue. We mentioned it. You have to have an official nucleus to call you a eukaryote. EU. What's EU means? Normal. So eukaryote means normal nucleus. Remember it like this. Eukaryote, normal nucleus. Prokaryote, before that. So you do not have an official nucleus that have a cell membrane. This is the most important part. Nuclear membrane. You have it, you're an official nucleus, I call you eukaryote. All right? Nuclear membrane. So if you remember from the prokaryote, from the bacteria, which is a prokaryote, remember how we culture them in different types of cultures that have the essential materials for them to grow and reproduce, right? This is not the case for the virus, because the bacteria needs the nutrients, right? It doesn't need a cell to reproduce. It needs just the nutrients. Give them what they need, and that's good enough, right? But for the virus, this is not the case. They have to have a cell. So you have, in order, if you wanted to culture them, if you wanted to grow them, you need a host, and this host have to be a living host. Okay? Living host means any type of cell, and this would be called the growth medium. So this can be animal. You get an animal in the lab and you inject it with virus. It will go into the cells, so that's good enough. Eggs, okay, any type of cell, whatever it is. We will see more details about that, but it has to be a cell, okay, and the cell is going to host that virus, okay. Again, still introduction, there is something else that's called a prion. And the prion is more simple than the virus. It's just protein. Okay? A prion is a protein. So we don't have genetic material? No, we don't have genetic material. The virus is, what are the two components again? Protein and genetic material. Yes, genetic material in the middle and protein surrounding that, right? This is just a protein. There is no genetic material. So we do not call it a virus. It's more simple than the virus. So we call it a defective protein. Defective protein, just a protein that's defective, but it can cause disease. So we consider it a pathogen still. Okay? And this prion can cause a couple of things that we need to remember, including the spongiform encephalitis or encephalopathies. What's encephalo and what's pathy mean? Yes, encephalo means brain, pathy means disease. So spongiform encephalopathies means disease of the brain in which the brain will look like a sponge, spongiform. Okay? We're not going to go into the details. This is not pathophysiology. Okay? We're just focusing on the virus itself. But it just go to the brain and it will cause spongiform encephalopathies. The two examples, just as an example to remember, mad cow disease and the Creutzfeldt-Jakob disease. These are two diseases that both of them are prion that's infecting the brain, causing spongiform encephalopathies. So can you tell me what are the two types of encephalopathies that you know? Spongiform encephalopathies, mad cow disease, and Creutzfeldt-Jakob disease, Creutzfeldt. Okay, so these are examples of the spongiform encephalopathies. Next, we talked about the virus in the very beginning, and we said this does not belong to any of the kingdoms, right? It's none of those. It's not bacteria, it's not an archaea, so it's not a prokaryote meaning, and it's not a eukaryote either. So what is it? There is a discussion about that. So they said, is it alive or not? How can we put it under, differentiate, or how can we put it under one of these kingdoms? It's none of those. So is it alive or not? There is a debate because there are some criteria of the virus that say it's not alive, and other criteria will say no, it is alive. Okay? What will tell us that it's not alive? It does not have a cell membrane, right? There is no metabolism. There is no growth for a reason. Remember we said that the virus is an obligate intracellular parasite, right? It goes into the cells and then what? And just duplicate. It does not grow. It does not become bigger, right? So there is no growth. How can you call it alive? There is no reproduction. It does not reproduce. It does not grow. It cannot even do metabolism. And metabolism is building and breaking. It's all these chemical reactions. It does not even have these enzymes to help them. Okay? So all of this combined will say, okay, this is not alive. But on the other hand, it's made of organic molecules. Isn't it? Isn't the protein coat is an organic molecule? It is. Does it have a DNA or RNA, which is the genetic material? Yes, so it is alive. When we see something that have genetic material, it's alive. And there is a purpose and life cycle that we will talk about. It's living for some reason. It go from cell to cell, go to the cell, infect the cells, replicate, leave the cell, go to other cells. So it does have a purpose, right? And there is a life cycle that we'll talk about it. So it is alive, it's not alive. They say, well, it's not completely, we can debate about that, but remember the criteria that tells us both of those. So again, being an obligate intracellular parasite, it will live into any type of cell. Like what? Like anything. Anything. And what is a cell? A cell is something that have a membrane and genetic material, right? Always remember this. In order to say it's a cell, you have to have a membrane and genetic material. If this genetic material surrounded by a membrane and a nucleus, you call it? What do you call it? If this is a cell, so it have a membrane and it have a genetic material. This genetic material is surrounded by a nuclear membrane. What do you call it? Eukaryote. You call it eukaryote if you have a membrane. If you still have a cell that have a membrane and it have genetic material but not surrounded by a membrane, nuclear membrane, you call it prokaryote, right? Does it infect prokaryote or eukaryote? Anything. Anything. It infect anything that is considered a cell. So you can say bacteria, which is a prokaryote, that's okay. Protozoa, fungus, algae, plants, animals, humans, we count under in biology with animals. So animals and humans, plants, algae, fungi, protozoa, anything. Basically anything that is alive, any cell, whatever it is. Okay? The thing is, this virus outside of the cell is not active. It will not become active until it get into the cell, and that's why we call it obligate intracellular. Okay? Now the structure. We said that it is a genetic material inside and a protein coat outside, right? We said that. And sometimes it have an envelope, sometimes it does not. Okay? Are we following so far? Okay. So genetic material is in the middle. This is the nucleic acid. This is inside. Outside protein, we call it a capsid. Okay? So you call those together nucleocapsid. Nucleocapsid. So the virus is genetic material and protein around it, right? Genetic material is inside, protein coat is outside. Genetic material is a nucleic acid, DNA or RNA, one of those. Okay? And surrounded by protein, we call it capsid. Since you have a nucleic acid inside and you have a capsid outside, I call that nucleocapsid. If that is it, that's it, you call it a naked virus. It's just a nucleocapsid. Are we following? Nucleocapsid. So this we call a naked virus. If it is covered by an envelope, we call it enveloped virus. And this will make a difference later on when we discuss the replication, which is very important. Okay? So is it enveloped or not? If it is just a nucleocapsid, we call it, if it's just a nucleocapsid, what do we call it? Naked, yes. If it does have an envelope, we call it enveloped. Okay, so far? If it have an envelope, it's usually have extensions, and these extensions are called spikes. And we will see the importance of that. Okay? These spikes will act like a key. Remember in the beginning, we said the virus go to the cell, it start to recognize that cell, and will get into it somehow, right? We didn't discuss that yet. But the question is, how this virus know what is this cell? By these spikes. See these spikes? Looks like a key, goes like this. This is a cap, this is the spikes outside surrounding the whole envelope, and go like this. They have something in that cell. So it goes like this. Let me see. This cell, you can fit in. Okay, this is my target. and it go to another cell. Let's say it looks like this. It try to fit in, no. Okay, this is not my target. It go to another one, no, no, yes. So this is how they recognize. It acts exactly like a key. So the spikes on the envelope, this is the key that will tell the virus, this is your target or this is your host or not. Okay, so the spike is for recognition and attachment. Recognition and attachment. Okay, so far? For the genetic material, which is a nucleic acid, it can be DNA and it can be RNA. And that's later on will give us the classification of the viruses into DNA virus and RNA virus. Obviously, if you have genetic material as DNA, you call it DNA virus. If you have genetic material as an RNA, we call it RNA virus. There is not a virus that have both of them. Okay? You're either DNA and you have only DNA, or you're RNA and you have only RNA. You cannot have both of them combined. Okay? There is no virus that exists that have both of them. Properties of the virus. Again, the virus is an obligate intracellular. It has to live inside the cell, and it's going to multiply. Does it grow? Does the virus grow? No, it does not grow. But does it replicate? Yes, it does replicate. How is it going to replicate and multiply? It's going to get inside the cell somehow. This will come in details, and this will be the activity for today. I will talk about it later on again. You're going to draw a cell and go step by step and tell me, we didn't discuss it yet, we will. And you will tell me, using these cells and the virus, here is the virus, recognizing the cell, entering into the cells and so on. You will draw that, and this will be today's in-class activities. We will talk about it when we do the replication. But anyway, for now, the virus is going to replicate, but does not grow, right? How is it going to replicate and why it does not grow? Here is how. It will get into the cell. We'll discuss the details. And it will go to the nucleus. It will take over. Okay? It will take over the nucleus. And it will order the nucleus. The contents of the nucleus. What do we have inside the nucleus? Any nucleus? DNA plus or minus RNA. So we have the genetic material. So the virus, once it goes into the cells, it will target the nucleus. It will go inside to the nucleus. It will take over. Okay? So you're not free anymore. The cells are not free to do whatever they want. I take a control. I occupy this nucleus. You are going to do what I'm telling you to do. Okay? You are going to make basically copies of me. You make copies of me. Okay? So this is replication. And again, we will discuss it. But the thing is, why not? Why you cannot live by yourself? Why you have to get into the cells? Why you do not grow? You do not have, the virus does not have enzymes. Okay? And it does not have the machinery for synthesis of protein. Can you tell me what is that machine that makes proteins? Ribosomes. Does it have ribosomes? No. Does it have enzymes for metabolism? No. That's why. You do not have one of those, so it cannot replicate by itself and cannot make its own protein. Okay? Size of the viruses is extremely small, less than 0.2 micrometers. It's extremely small. They call it ultra microscopic, not just microscopic. So if you have a regular microscope, like the light microscope, you can't see it. You cannot. You need an electron microscope. That's how small it is. Remember the microscopes? We talked about this, right? The different types of microscopes. The one that have the highest magnification is the electron microscope, and this is the only one that can see it. Okay? So this is just showing you the different sizes. Bacteria, big bacteria, small bacteria, and giving you the viruses. It's too tiny to the extent of you have to use electron microscope. So can you see the virus with the light microscope? No, you have to have electron microscope. So, does the virus have ribosomes? No. Does it have the enzymes needed for metabolism? No, it does not. That's why it needs to occupy the cells, and it does have the minimum. It does have the minimum. What's the minimum? The genetic material and the protein. This is the minimum that helps them to invade and take over and control. So again, the core of the virus is genetic material. It can be DNA and RNA. It has some enzymes in the matrix, a little bit of enzymes in some viruses. Okay, just a little bit, a couple of enzymes. That's not all the enzymes needed for metabolism. It does have some, and this is the central core. And the outside, what do we call the surrounding of the genetic material? Protein capsid outside. And some viruses have envelope and some viruses does not have envelope, right? Now the capsid. The capsid is made of units that called capsomeres. Capsid, capsomeres. Okay? Look at these units here. This is the capsid outside all of these units. One unit is called a capsule. Okay? When you have the nucleic acid together with the capsid, we call that nucleocapsid. Okay? Why do you have the capsid? Why don't you just have these genetic material? Every single virus, you have to have those two components, or you do not consider it a virus, right? You have to have those two components. Nucleic acid in the middle, at the center, surrounded by a protein capsid. Why do you have a protein capsid? And why some viruses have an extra envelope? The protein capsid is serving two purposes. Number one, it's anchoring and protecting the genetic material inside. The other thing is it's important for recognizing and attaching, right? Now, if you have like genetic material like floating around like this, it will not be able to attach itself and occupy the cells. Okay? Are we okay so far? So why do you have a protein capsid, plus or minus envelope? Recognition, attachment, and to anchor, okay, the genetic material. Okay. Now a little bit more about, yes, yeah, of course.
Sorry, so what's gonna be the difference between the capsid and the spikes on the capsid, on the envelope?
The capsid is just a protein that's surrounding the genetic material, and this is every single virus on Earth. You have to have genetic material in the middle, and you have a capsid surrounding it. Okay. If you have an envelope, this envelope will have these spikes, and this will help you to recognize. If this is a naked virus, so you don't have this envelope and you don't have capsids, right? You don't have spikes. The capsid itself will recognize the host. Okay? You're welcome. Obviously, if this is an envelope and you have spikes, that's a plus, right? This is an additional tool, act like a key, so it will be easier to recognize the host. It will be easier to attach, but the capsid itself can attach. You don't have to have an envelope to attach. Okay? Now, depending on how the capsid looks like, it can be any one of those shapes. Okay? Icosahedral or icosahedron. It can be polyhedral. It can be helical. It can be complex. So, icosahedral or icosahedron means icosa is a prefix that means 20. Okay? And hedron means face. So this shape have 20 different faces, which is triangles basically. And you have 12 corners. We call that the icosahedral. If it looks like this, like a spring like this, we call it the helical part, or the helical shape. If it just looks like this, you have multiple, not necessarily 20, we call it polyhedral. Okay? What if you have both of them? Like you have this and this together. This is polyhedral or icosahedral, and this is helical, isn't it? This looks like this, and this looks like this, right? You call it complex. Okay? So according to the shape again, icosahedral, polyhedral, helical, complex. Is that okay so far? This is depending on the shape of the capsid. Now there are some viruses that we call it atypical virus. Okay? Atypical means we have typical and atypical. What's A means when you put A in, as a prefix? Not. So typical virus is what we just talked about so far, right? Any typical virus on Earth will have the same thing. Okay? Genetic material in the middle, DNA or RNA, surrounded by protein capsid, right? Plus or minus what? The envelope. You can have the envelope or not. This is, we call it the typical virus. Every single virus is like this, except this is atypical. Something like pox virus. They do not have that typical capsid that we talked about. The capsid here is lipoprotein. So what's a regular capsid? It's just a protein, right? This is lipoprotein, so it's not typical. The other one that's also non-typical is called bacteriophage. Bacteriophage. And bacteriophage are also atypical because it does not look exactly like what we talked about before. It does not exactly look like a genetic material with surrounding capsid. And the other thing that's unique about those is come from the name. Bacteriophage. Bacterio means bacteria. Phage means eaters, right? These are the bacterial eaters, the bacterial invaders. They invade the bacteria specifically. They do not invade us, ourselves. They invade the bacteria. They are very specific. And also, they have these attachments. They have tails here, and these are called the attachment fibers, which is not the case for any other typical viruses. Okay, so far? So we have typical, we discussed it. Now atypical. You have pox virus and bacteriophage. Okay? So again, if you have the envelope outside, we call you an enveloped virus. If you not, we call it naked virus. Okay? So the one that have envelope, these are mainly animal virus. Remember in the beginning we said the virus is an obligate intracellular. They have to live in any cell, whatever it is, including anything, including bacteria, protozoa, plants, animals, right? Specifically the animal ones, these are mostly the enveloped one, they are mostly the animal viruses. So the virus that invade animal, most of them will have an envelope, okay? And they get the envelope, like if you ask yourself, where did you get that envelope from? Okay, they get it when they leave the host cells. After invading, after replicating, we will discuss all the details. But while you're leaving the cells, you will steal part of the cellular membrane and you make it as an envelope for yourself. And you will add to it spikes. So when do they make the envelope? While leaving, release, while leaving. After they've done everything, I replicated, I duplicated myself, I made different copies, now I'm leaving. Wait a minute, before leaving, I will get an envelope. These steal part of the membrane, of the cellular structure, and they make an envelope out of that. So here is the structure again. Inside here, this is the nucleic acid, DNA or RNA, and around it you have the protein coat, and you have an envelope that have spikes. This brown here is a matrix that have some enzymes. Okay? So what was the function of the capsid and envelope? attachment, protection, anchoring, recognition. It's around that, okay? You get to know your host, you recognize your host, whatever you want to call it, and then you attach yourself to it. This is the function of both capsid and envelope. If you have an envelope, it will serve that purpose. If you don't, the capsid will do it. In both cases, it's for protection, anchoring the genetic material, and for recognizing and invading the cells. These are just some examples. So some viruses will have, when you call it icosahedral virus, what are we talking about? Icosahedral is for the shape of what? For icosahedral. This is the shape of what? Which part of the virus? The capsid, yes. So these are the ones that, and what icosa again? 20, yes. Icosahedral means 20 sites. Icosa means 20. So rotavirus is an example of icosahedral virus. Herpes simplex is also an example, but rotavirus is naked and herpes simplex is enveloped. These are just examples. Examples of naked viruses that have a helical capsid. Plumpox virus, this is just for plants, but it looks like this. They have a helical capsid. Poliovirus is an example of virus that's icosahedral capsid, no envelope. For the enveloped ones, the ones that have helical nucleocapsid is like mumps. Icosahedral is like herpes and HIV. Complex is both icosahedral and helical at the same time, right? Part of it is icosahedral and the other part is helical. And the example of that you need to remember is pox virus. Okay, so pox virus is an example of complex virus. So we said that the genome can be DNA or RNA, and this will make a difference to differentiate the different types of viruses. So it can be DNA viruses and it can be RNA viruses. If it is DNA, remember in AMP, the DNA is double-stranded, isn't it? Sometimes it's not, but the vast majority is double-strand. Okay? Most of it is double-stranded. If it is double-stranded, we call it DS. DS, double-stranded, right? Occasionally, the vast majority is double-stranded, as we learned before, right? DNA is double-stranded. The D goes with the D. DNA, double-stranded. But sometimes it might be single. This is an exception. And you call it SS, single-stranded. And the way it's arranged or looks like, it can be circular, like the top one. It's double-stranded, as you see here. See the two strands, red and blue? And it looks like circular structure, or it can be linear like this. Okay? If it is RNA, RNA is usually single or double? Single, right? But occasionally it might be double. So the rule is, DNA is double, RNA is single. And there are some exceptions. DNA, sometimes it can be single, RNA, sometimes. And the virus, it can be double. And sometimes it's even RNA. If it is RNA, sometimes it can be pieces, different pieces. Now, if it is RNA, here is the problem. Do you guys remember when we, in AMP, I don't know if you remember or not, but in AMP, remember the DNA is the manager, right? The RNA is the helper. The DNA is the manager that have the actual code, right? This is going to manage the process. Remember this? DNA makes messenger RNA, leave the cell, go to the ribosomes, direct the protein senses in the ribosomes, right? Transfer RNA is getting, so RNA is not the manager, right? It's not managing. DNA is a manager. So if this is a DNA virus, everything is okay. Your DNA, you take control of the DNA of the nucleus, so you're fine, right? You're ordering the RNA to do this and that. What if you are an RNA virus? It depends. This is something unique about the virus, that the virus, if it is an RNA, there are two types. If you have an RNA, there are two types. One of them that have the ability to do the translation, to take over and do the translation, and we call that the positive sense RNA. Okay? The other type is called the negative sense RNA. It cannot. One more time. The viruses can be DNA or RNA. If you're DNA, you're fine. DNA is a manager, right? It can direct the protein synthesis, so that's fine. What if you are an RNA? This is an issue, right? Well, it depends. It depends. Some RNA can manage for the viruses. It can act as a genome and act as a translator at the same time, like a messenger RNA, and some others cannot. If you can, I call you the positive sense. So if you're a positive sense, you're fine. You can act kind of like the DNA virus. Is that okay so far? What if you are a negative sense? You cannot. You cannot. You cannot direct the protein synthesis. You cannot replicate yourself. What are you going to do? We will talk about this, but simply these viruses, like HIV for example. HIV is one of these negative sense RNA viruses. So how can you, if you are a negative sense, RNA, not just RNA, you're negative sense, you cannot direct protein sense. What are you going to do now? They are going to utilize enzymes inside the cells. There's something called reverse transcriptase. Do you remember transcriptase? Transcription is to make what from what? Do you make DNA from RNA or RNA from DNA? Transcription. RNA from DNA. We call that transcription, right? When you transcribe, like I have this DNA, I make messenger RNA. This is called transcription, right? But this is a normal transcription. Sometimes some enzymes can do this. Reverse transcriptase. You get the RNA and you convert or you make DNA out of it. And this is their way. Later on when we start to go to the treatment and stuff like this, we will be talking about different steps in the treatment, including reverse transcriptase inhibitors. So you will see some medications later on, like AIDS. How do they try to treat AIDS? At least usually the treatment, unfortunately, the treatment for AIDS is not is very complicated, but at least you can slow it down, right? Reverse transcriptase inhibitors. This is not pharmacology, I'm not going to go into the details, but basically, if this is a negative sense RNA and I stopped reverse transcriptase, can you take over? You cannot, right? You're a negative sense, you cannot take over. You're trying to use your reverse transcriptase. If I stopped it, you cannot take over. Is that clear so far? Any questions? We're fine? Okay. What are the enzymes that are needed for the virus to do the replication? Polymerase. This is what's helping you to do DNA or RNA. What does that mean? It means DNA polymerase and RNA polymerase. You can make copies of yourself and you can make RNA out of the DNA. Replicase is to replicate RNA, basically. What's replicate means? Make copies, yes. So this is replicase. Reverse transcriptase is just what I mentioned. It's a transcriptase, but the opposite. Okay? You're making RNA into DNA. You make DNA out of the RNA, which is the reverse, the opposite, right? HIV is an example of that. Now, classification of the viruses. How do you classify viruses? Structure, and we talked about this. Are you icosahedral Are you helical? Are you complex? Are you naked? Are you enveloped, right? This is one of the classifications. The chemical composition, you have DNA, you have RNA, what kind of enzymes. This is the chemical composition. The most important of all is genetic makeup. This is the most important, okay? Tell me about the genes that you have, exactly what kind of RNA and DNA do you have. I will tell you the classification. You start with orders. If you talk about the virus, you start with orders, going into families, and then the geners, and then you go to the species. So orders, we have 7 orders, and then we have families. When you talk about families, you call it the viridae. Okay, viridae. Viri is virus, ed means family, just a translation. Okay, what's viridae? This is a Latin. Vir, the first part, means virus. It's a prefix. Okay, ed, this means family. So viridae means the virus family. So this is the name of the family. This is important. Okay, this is going to help us to put the names or to understand the names. And then after the families, you have the genera, which is, it's always italicized. Both of them are going to be italicized, families and genera. This is how you will see it. Look at this. Isn't all of it is italicized? It has to be both family and genera, not the order. Are we following? We have order, families, genera, right? Orders, that's normal. Both families and genera, both of them has to be italicized. If this is a family, you have to put viridae at the end. Are we following? Okay. If it is a genera, you put virus at the end. So look at this example. Herpes viridae. What is this? The name of what? Family or genera? Family. Simplex virus. It ends with virus? Genera. Italicized. Both of them are italicized, right? So this is how you make the name. Family and genera. Okay, can you answer this question?
False.
What do you think?
False.
False? Why?
Because they contain both.
It cannot contain both, right? Either DNA or RNA. Okay, these are the steps of multiplication, and this is what I want you to draw. You get just a piece of paper, and you're going to draw 1, 2, 3, 4, 5, 6 cells, and you do a virus doing each one of those. According to your understanding, I have a video that I'm going to show you, so it will give you a better idea, and you just draw it and label it, okay? Why? Because I want you to really understand this, and I want it to stick in your mind, okay? When you draw something, you'll remember it. So what are the exact steps? One by one. You start with adsorption. What's absorption mean? Adsorb means to attach. Adsorption, to attach. So adsorption is to bind, the virus binds into the host cells. What is the virus using to bind?
Spikes.
The spikes if it is enveloped. What if it is not enveloped? Using the capsid, right? So you do adsorption. Next part is penetration. Penetrate into the cells. You get into the cells. Followed by uncoating. What's un mean? Without. Uncoating means you take the coat off. Uncoating. Okay? Meaning, what was the coat? It's the capsid. Just take the capsid off. Take your coat off. And then synthesis. You make the components. What are the components? The protein and the genetic material, DNA or RNA. Okay? So we are going to make these components. Assembly is when you put them together, like the assembly plant, right? Assembly plant means give me component number 1, give me component number 2, I put them together, and I make the virus. This is called assembly. And then the release is the last step. There are different ways by which the virus is going to leave. It can, if it is leaving, it's going to destroy the cells. Okay? I'm leaving, I'm going to destroy the cells, and all my copies will get out to find other cells. This is one way. The other way is, how about I leave the cell alone and just make a budding, an extension, exocytosis. And we will go through these details, but these are the steps. If you're following, what do you think will happen during release? What happened to the virus during release? It can or cannot, but what are the changes in the shape, in the structure? The get and the envelope. This is the step during which they will acquire the envelope and the spikes. Okay, so far? So step by step. Adsorption. This is simply either the spikes, if the virus have an envelope, or the capsid. One of them is going to adhere or attach or adsorb. Most of them are the same, right? All of these are the same. Adhere, adsorb, attach, it's the same thing. Whether you have an envelope or not, you can adsorb. It's just which part is going to help you to adsorb. To adsorb to what? Some virus to the host. So some viruses will infect one host. Some viruses can infect 2, 3, 4, 5. We call that the host range. Okay? The host range. How many hosts, or what are the hosts that you can invade? Okay? Which organ, basically? Can you invade one organ, 2 organs? How many species? Can you invade animals and humans? Can you invade also bacteria, and so on? So this is called the host range. Host range will depend on what? Simply on the cell membrane. So it goes like this. I'm the virus, okay? I'm the virus and I'm going around like this, and I go to this cell. Show me what you have in the cell membrane. Try? No, you're not my host. Okay? Go to the next cell. Let me see what you have in the cellular membrane. No, you're not my target. You're not my host. Go to this one. Fit in? Yes, you are my target. I will invade you. So what determines the host range? The cellular membrane. Not the viral envelope. The viral envelope, if it have a spike or something, yes, just the key. But which cells to invade? Which host? The cell membrane. Depend on the cell membrane. So here are examples. Hepatitis B. The host range is human liver cells specifically. They do not go to anything else. This is their target and this is their host. How did they know? Like, this is hepatitis, it got into the blood somehow and it's moving in the blood. Why it does not invade any other cells? The liver cellular membrane, like this. This is the liver cellular membrane, have this. And I have the spikes, for example. I go like this, yes, you are my cell. On its way, it can find the intestine. No, not my target, and so on. Host range. A poliovirus, for example, it invades intestine and nerve cells. For what? For primates. Any primates. Okay? Monkey, humans, any primates. Rabies, for example, any mammals. Isn't that a big range? It's a big range, right? Hepatitis B, human liver cells specifically. This is a very narrow host range, isn't it? Just human and liver cells. That's it. The poliovirus is more than that. Intestine and nerve for any primate. Rabies, do we know the rabies? What's the rabies? Come from what? From which animal? Dogs, specifically. It can be other animals, not just dogs, but this is the most common, right? They tell you, you be careful if this dog is not immunized and it bites you, you can get rabies, right? Rabies is coming mostly from dogs. Does it only invade us? No. It can get into any mammals. Next is penetration. You are going to penetrate. How can you penetrate? It depends. Are you enveloped or not? There are two different ways to penetrate. How can you get into the host? There are two different ways. If you are enveloped, you can do fusion or endocytosis. If you're naked, you don't have options. You have to do endocytosis. And what's fusion? Fusion. So fusion only if you are enveloped. So look at this. This is a cell, like this, and this is a virus, and here is an envelope, right? It will go to the cell, and how does it penetrate? It's just going to fuse itself like this, because it has an envelope, right? So the envelope, this is the envelope right here. It got fused with the membrane, right? And then it will move in, will be like this, and then we'll do uncoating. You end up having this, right? This is if you have an envelope. You can do this. The other type is, instead of fusion, this is a cell. Now if you are enveloped or not, this is naked, this is enveloped, right? Both of them can get into by endocytosis, okay? So basically the virus will eat you. It gets completely enter, goes like this. Just enter in. Okay? We will have a video for that, but just imagine what's happening here. So which one can do fusion? Envelope. Which one can do endocytosis? Both of them. Both of them. Because endocytosis, endo means inside, cytosis means cyto, cell. So endocytosis means I get you into the cells. It doesn't matter if you're enveloped or not. But fusion depends on the envelope, right? And then you're going to do the uncoating anyway. So look at this. This is envelope. Get into. See how it's fusing? Because it does have an envelope. It will get in. What do you call this part? No, what do you call this unit? Both orange and blue. What's the yellow or orange? The genetic material. And what's the blue? The capsid. So this is nucleocapsid. Okay? Where is your envelope? It's left outside. It's fused. So now you have a nucleocapsid. When you release the genetic material, we call that uncoating. So what's the coat? The capsid. The coat is the capsid. When we say uncoating, means take off the capsid. So now you have a free, in this case it's RNA, but it can be anything, and this is free to take over. The other thing is, whether you are enveloped or not, it's just the cells will make a cavity for you and detach part of the membrane, take you inside and release. Okay? Next is going to be synthesis. Synthesis of what? The two components. What are the two components? Nucleic acid and capsid, right? So replication is to replicate yourself, to make copies of yourself. To make copies of what? Of both components, the nucleic acid and the capsid. Nucleic acid, this is the DNA. The DNA is going to replicate yourself. And how to make the proteins? Ribosomes, right? You send messenger RNA to the ribosomes to make proteins. So in order to replicate, here's what I'm doing. I'll go to the nucleus, the DNA. I'll control the DNA and I'll tell the DNA make copies of me. Okay? So now I have the nucleic acid part. And then I send messenger RNA to the ribosomes, make copies of my capsid, protein, right? And then when I put them together, this will be a new virus. Okay?
So when the encoding happens, does the genetic material gets injected into our nucleus?
Yes. Yes. It take over, basically. It go to the nucleus and take over the nucleus. Occupation. It go to the nucleus. Now I'm the master, and now I'm going to tell you what to do. And here is what I want you to do. My goal is to make a lot of copies of me, but go step by step. You are going to do what I'm ordering you to do, okay? Step number 1, DNA, make copies of me, of my genetic material. So I have the genetic material now, right? Then I will also tell the DNA, send messenger RNA to the ribosomes to make this type of protein, which is my capsid. Now I have the 2 components, right? Multiple copies of my gene, multiple copies of the capsid, right? Put them together, which is called assembly, which is the next one. Then I have a lot of viruses. So assembly is the next part, which is you put the nucleic acid. I have multiple capsids, which is proteins. I have multiple nucleic acid, which is genetic material. When I put them together, we call that assembly. You assemble something, right? So I will have a lot of copies of the virus when you put them together, and it is going to leave at the end. Okay? So here it is. Look at this. You're taking over this. You're making copies of the nucleic acid. This. You're making copies of the capsid. Put them together. You're making new viruses. Multiple. Then you're going to be released. Do you remember when you entered, when you penetrate, there are 2 ways to penetrate, right? Fusion and cytosis. This one is kind of similar, but the other way around. The first type is, what did we call this again? So the first way is exocytosis. The opposite. The same way you entered is the same way to leave. Okay? So you're going to move and you're being ejected out. If that happens, the cell is intact. The cell is not going to die. Okay? So I occupied you. I will leave you alive. I'm not going to kill you. I'll take over, make copies of me, and then I leave. Okay? So the cells will stay alive. It's not going to die. The other way is called lysis. What's lysis mean? Burst, breakdown. So the other type is, no, I'm not leaving. I'm rupturing the whole cell and spreading around. So obviously the cells are going to die, right? So you have 2 ways. The first way is called exocytosis. You also call it budding. Okay? Exocytosis or budding. Budding means to make a bud, like exocytosis. You take a bud out, which is part of the membrane surrounding you, and I push you out. And this will happen only if you have an envelope. So the envelope virus can do budding or exocytosis. You need to remember both of them. Budding, exocytosis is the same thing. Budding, exocytosis. Okay? This is if you're enveloped. The cells are not going to die. If you are not enveloped, you don't have options. Okay? You cannot do the exocytosis. You don't have an envelope. If that's the case, the cells are going to die immediately. I'm leaving, but I'm killing the cell before I leave. Is that clear so far? Any questions? So here it is, the whole entire process. From entering, leaving, taking over, adsorption, penetration, uncoating, synthesis, replication and making the protein, Now you have the nucleic acid and you have the capsid, and then you leave. You leave by exocytosis budding or lysis. Okay. Bacteriophage. Bacteriophage means bacteria eaters or invaders. They invade the bacteria. They do not invade us. Yes.
So for the release, so since it's envelope, what does it gain? Because you said it was an envelope. Does it gain a new one afterwards?
Yes. Yes. This is a good point. Yes. Remember always during release, if this was an enveloped virus, while you're entering, didn't you leave your envelope outside? And you entered in, right? When are you going to regain it? You take it again during release, which is exocytosis, body, right? If you're leaving, you reacquire the envelope again because it was an envelope virus. Okay? So bacteriophage are the bacteria invaders, the invaders for the bacteria, the phages for the bacteria, and the invade bacteria. E. coli is one of the most common ones. E. coli is a bacteria that can be invaded by different types of viruses, DNA viruses. It does the same steps of the animal virus like we talked about. Do you remember the steps? This is very important, and you're going to draw it. I will show you some videos as well. But the steps, can you tell me the steps? What's the first step? You go and absorption, penetration, uncoating, replication, synthesis, assembly. What's after assembly? Release. Penetration was in the beginning. Release, right? The exact same steps. Happen with the bacteriophage, but you're doing that instead of a cell, animal cell, you're doing that in a bacteria. Minus one thing. Here is the most important part. The most important difference. That the bacteriophage does not enter into the bacteria itself. That phage, which is the virus, it just injects the nucleic acid. It injects it, not the whole bacteriophage. It just go like this, absorption. Are you doing penetration, like getting into the cells? No, I'm just injecting my nucleic acid. Did you get the idea? So what you don't need in this case? What's the step that will be missing in that case? Uncoating, yes, exactly. Uncoating. Why do you need to do uncoating? You are just a genetic material, right? No uncoating. Okay? So if to ask you, what's in the bacteriophage that's not in the animal virus, and what's in the, I'm talking about replicating. Bacteriophage have a step that does not happen in the animal virus, and animal virus have a step that does not have in bacteriophage. What is the step that the bacteriophage have that does not happen in the animal virus? Injection. It injects genetic material. And what is the step that happen in the animal cells, not in bacteriophage? Uncoating. Okay, great. And if that's the case, it will lead to lytic only, lysis. Why not exocytosis? It did not get into the bacteria in the first place. It was just genes, right? Remember which one does the exocytosis or budding? Enveloped, right? Was that enveloped? This is not even the whole virus, it's just the genetic material, right? Okay. So here are the steps again: absorption, penetration, uncoating, replication, synthesis, assembly, maturation, then lysis and release. Now in this part, there is an additional thing that I need to talk about. And let me ask you again, in which step does reacquiring the envelope happen? If you are an envelope virus. Exocytosis or release? Release, exocytosis. Okay. Now they noticed something here that's important to understand. In all what we talked about so far, while the virus is about to be released, there is an additional step that can happen. So when you release, if you are not enveloped virus, okay, and you're going to be released, not enveloped, isn't that lead to lysis? Not enveloped, right? No body, it's not exocytosis. You're going to release and the cell will die, right? Here is something that's different, and they call that the lysogenesis. Lysogenesis. Instead of lysis, lysogenesis. Here's what's happening. It's something interesting that's different, completely different. All these steps that we talked about is okay, but here's what's happening. Absorption, penetration, okay, assembly, everything. But instead of doing the assembly and leave, no, you're going to incorporate yourself into the host DNA. Incorporate yourself. I'm not going to do like the traditional ones. I'm not going to put my components together, assembly or assembly, and then leave. I'm not going to do that. Here's what I'm going to do. This is a special case, okay? And we call that the lysogenic, not the lytic. Lysogenic. Are we following? Lytic is just a regular way. Lysogenic is something different. What's the lysogenic? I'm not going to kill you. I'm not going to do anything. Here's what I'm going to do. Did you make copies of me? Okay, thank you. You did my copies. Everything is good. Replication, all of that. I'm not leaving. I'm not going to kill you. I'm going to incorporate myself into your DNA. And I'm not going anywhere. Why are you doing that? Here is why. Because human cells in general are going to duplicate. Remember that in any cell? Remember that we have transcription, you make copies and all of that, right? When I incorporate myself into your DNA and you are replicating yourself, you're going to replicate me automatically, right? Isn't the DNA of the cells and RNA, you make copies all the time? Anytime you make copies, automatically you're making copies of me. Okay? So we call that the lysogenic cycle. And this is specifically in chickenpox, herpes simplex, and HIV. They do that. Yes?
So is that like the same as like the dormant and opportunistic diseases that we have? Like how HIV states in there and then you see like the cold sores and they have like a flu or something? Oh, that's what it kind of
This is latency. If you're talking about the herpes and cold sores, this is called latency. We will come to that point. But part of herpes simplex or chickenpox or something, the way they go into the cells is the same way, but they don't leave. They wait some time until you make enough copies. Not the same way of making copies, make nucleic acid, make capsid, put them together, leave. No. I will make more and more and more copies by incorporating myself. At the end, I am going to leave anyway, but not now. I'll go to lysogenic first. And chickenpox, herpes simplex are good examples for that. Look at this. What is this? What is it doing here? Injecting. So what is this? Bacteriophage. This is bacteriophage. How did you know that this is bacteriophage? Because it's injecting. You see the red injected? Not the whole virus, right? Not even the capsid, just injection. So this is obviously a bacteriophage. Okay, so here is a video. Not this. Okay, the sound is not. So this is lysogeny that we talked about, or the lysogenic cycle, which, as we have seen in that video, that is incorporating itself into the host DNA or the host genetic material. It's going to be replicated, it replicates itself while the cell is replicating itself. It also replicates the viral genome, and it's doing more and more and more and more copies until something happen to trigger the lytic cycle. So lysogenic cycle is ultimately going to switch into the lytic cycle. Just this name is important to remember: temperate phage. Temperate phage are the phages that like have a temper. The phages that wait. They do not lyse the cell. They incorporate themselves. So you call those the temperate phages. So temperate phages are these types of phages, or the viruses, the viral DNA phages that incorporate itself. We call the temperate phages. Okay? So it's going to produce something called the prophage, and prophage, you know, pro means it's not active yet, usually. Pro means inactive yet. And then it will become activated and become enter into the lytic cycle. Okay? So this is lysogeny, which is replication without killing the host cell. How? By incorporating yourself into the host DNA. Okay? Here is one more thing that's also important. While doing this, while incorporating yourself into that cellular DNA, some viruses, remember these three names: diphtheria, cholera, and Clostridium botulinum. These viruses have something unique. After incorporating themselves, somehow, for some reason, they order the DNA to make some toxins. Okay? So these are called toxogenic. They make some enzymes and toxins, and they call it the lysogenic conversion. So while you're doing that, while incorporating yourself, you order the chromosomes to make toxins for you. So these are called the toxin-producing bacteria. So how the diphtheria and cholera and Clostridium, how did they make toxins? The virus told them to do that. The virus, which is the phage. So later on it can, or ultimately it's going to switch, activate and switch into the lytic cycle. The temperate phage is a phage that's temperate, means it's not going to destroy the cells. It's temperate. It's going to get into, like in this case, into the bacteria. It's going to take over, incorporate themselves into the DNA, and then it's going to, while the bacteria in this case is replicating itself, it's automatically replicating that virus that's incorporating itself. So in that the case, whether the cell is bacteria or not, all of these are cells, it's not killing the bacteria. So the virus, the phage, the virus, in this case it's a phage, that's called the temperate phage, is the one that get into the lysogenic cycle. Okay? So which virus go to the lysogenic cycle? The temperate phage. Is that clear? You're welcome. Okay, can you answer this question for me? Not in bacteriophage uncoating. Yes. Are we good so far?
Yeah.
This slide is extremely important. Very important. You have to know it. Okay? First of all, host range, we talked about this. What determined the host range? Huh? The cell membrane. Receptors on the cell membrane. Okay? The protein or the envelope of the virus will fit into it to know if you are my host or not. This is a host range, right? Simply, I have my key. Any cell that have a receptor that fits into my key is my host. 1, 2, 10, it doesn't matter. Okay, this is called the host range. Here is something that's about the host range. Flu, the host range for the flu is the lung. Okay, the respiratory system and the lung. They call it pneumotropic. Pneumo means lung. Pneumotropic. Chickenpox, the target is skin. The host is the skin specifically. They call it dermotropic. You know what tropic means? Feeds into, or live in. So dermotropic means tropic in the derms, meaning it invades the skin. Okay? So flu is pneumotropic. What pneumotropic means? Lungs. So, or the respiratory system in general. Respiratory system. Chickenpox is dermotropic. What does it mean? Skin. Hepatitis is viscerotropic. Viscera, yes, which is organs. Here it's the liver. They call it viscerotropic. Okay? Viscerotropic means it invades the viscera. Which viscera? Liver. In this case. Encephalitis is neurotropic. What does it mean? The neurons. Yes, the brain or the neurons in general. Brain and spinal cord. Okay? Is that clear? Now, the cytopathic effects. Cyto means cells. Pathic means disease, or pathology means disease, right? So what are the changes that will happen into the cells because of the occupation or the entrance of that virus? This is very important to remember. Okay? What exactly is going to happen? The cytopathic changes. It's going to alter the DNA. This is one of the effects. It doesn't have to be one. It can be a lot, more than one. Okay? It can alter the DNA. This is one. If it go into the lytic cycle, it's going to cause cell lysis, isn't it? It can do some gross changes in the shape or the size of the cell. Like if I occupy this cell and I want you to make more copies, and these copies are big, I will make this cell to become bigger in order to accommodate my needs, right? Or I can make it look different. This is one of the changes. It can make other changes inside the cell itself. Like it can make multinucleated giant cells. We call it syncytium. It can also make inclusion bodies. These are all changes. Like it will make the nucleus of the cells to become multiple nuclei. Are we following? We call that multinucleated giant cells, also known as syncytium or syncytium. What's a syncytium? Multinucleated giant cell. A big cell that have multiple nuclei. Is that clear? Can that happen to the cell? Yes. The viruses can do that. It can also do some inclusion bodies inside the cytoplasm. It can make different bodies to contain something. They all depend on the needs of the virus. So it's basically when I occupy you, I do whatever I want. I can kill you, like it, lick it. I can change your DNA. I can change your shape, right? I can make you bigger. I can change your shape. I can make you multinucleated, right? All these changes can happen. Some changes are more specific, like rabies. Rabies infects the neurons, right? If somebody has a dog that's biting them, you have to go to rush to the hospital, and they will tell you have to take the vaccine like every 3 days, whatever they call it, or for a week or something, right? Why do you have to do that? Because the target is neurons. This is the target, right? If they actually go to the neurons, they will create dark colored bodies that are called negri. Negri, what's negri means? Black. You call the negri bodies because these are black bodies that's inside the neurons. So what are the changes that happen in the neurons if invaded by rabies? Negri bodies. Negri bodies. Another one that's more specific, mononucleosis. Infectious mononucleosis. Mononucleosis is a virus that invades lymphocytes. Specifically, they invade lymphocytes. This is the host range. Okay? And by doing that, they make the lymphocytes swell, and they call it the downy cells. The downy cells. What are the downy cells? Swollen lymphocytes by infectious mononucleosis. Is that clear so far? If you have any question, let me know. Now, is the host cell is going to die by going into the lytic cycle or not? Sometimes it doesn't die immediately. The cells cannot die. Sometimes an infection can stay for weeks or lifetime, and it goes into different cycles. And this is called the latent state. Somebody was asking about this. It goes into, we call those the latent virus. What's a latent virus? Here's a latent virus. Herpes simplex. Herpes virus. Somebody was asking about these cold sores, right? Cold sores. Here's what's happening exactly. Herpes simplex gets into your mouth somehow. You got infected from somebody else in a different touch, kisses, whatever it is. You got the virus anyway, okay? The virus will go to the mucous membrane. It goes inside. Are we following? It will go into the sensory neurons, and it will move inside the sensory neurons until it goes to a ganglia. Do you remember the ganglia from ENT? A collection of cell bodies. Once they find the ganglia, because this virus is weak, so it goes to the ganglia and will say, okay, I'm staying here for some time. I'm not strong enough to do all these lytic cycles. This is a weak virus. So it will go to these ganglia and will say, okay, I'm staying here. I'm not doing anything. Okay? I'm waiting. Until you are weak enough, because this virus is weak, right? If they try it right away, they cannot do anything. So, and they know themselves. I'm weak, I can't do anything. I'm going to hide into the ganglia and stay there. How long? I don't know.
So this is the temperate phase. When you stay there?
The temperate phase, it can be one of the temperate phase to get into that latent virus. Yes, you're right. So that latent virus can become chronic. In this case, it will become chronic. How? Exactly like I'm saying right now. It goes into your mucous membrane and skin. Find the way to the sensory neurons. Go step by step into the sensory neurons until it finds the ganglia. And the ganglia is going to stay. How long? I don't know. Until you're weak enough. I cannot do anything. Are you weak? What do you mean weak? Weak is like you're not eating good, malnutrition, you're having like flu for some time, aren't you busy fighting? So you're kind of weak, right? If that's the case, they will say, okay, even though I am weak, but you're weaker than me. Now there is no need for me to stay in the ganglia. I'm leaving. I'm going backwards. I'll go to where I entered in the first place, which is your skin and mucous membrane, right? And then I'm going to replicate and do that cold source. Did you get the idea? Once you regain your immunity back, like you are taking cortisone, which make you weak, you're not eating much, you're weak somehow because you just you had flu or something, and then you are recovering. I'm eating good, I stopped the cortisone, I don't have the flu anymore, you're regaining. They will say, oh, you're getting stronger, I'm going back to the ganglia, I'm going to hide again. And it keep going into that. Inactive in the ganglia, when you become weak for some reason, the immune system going down, I'll reactivate and go and infect you, calling the cold source. You regain your immunity, I'm escaping, going back, hiding, and so on. This is called the chronic latent phase. Chronic latent phase. Does it go away? No. Unfortunately not. Does not go away. And that's why you can ask yourself, like, why I'm getting the cold sores, and then it disappeared for a couple of months, and then it come back, and so on. You didn't kill it in the first place. It was hiding. You didn't kill it, right? It stay with you sometimes for life. So this was the herpes simplex. Same thing for the herpes zoster. It does the same thing, but not here, not in the mouth, in different other spots. Exact same thing, exact same scenario. Measles, on the other hand, they do the same thing, but they hide in the brain. Is that clear? Measles, herpes simplex, herpes zoster, all of them do the same thing. They are chronic latent. They hide. But where do they hide? Herpes, the two types of herpes, they hide in the ganglia. Is that clear? Measles, they hide in the brain. Okay? Some viruses, they call it the oncovirus. What's onco means? Cancer or tumor. Yes, onco is a prefix that means cancer or tumor. So what genic means? Genesis, generating. So oncogenic means generating cancer. So some virus can generate cancer? Yes. Animal viruses, some of them. How do they become oncogenic? Do you remember the cytopathic that we just talked about? Wasn't one of them is to alter the DNA? Yeah. If they do that permanently, like you change the DNA, is that something that will be corrected or not? If it is corrected, that's okay. If it is not corrected, it's a permanent damage of the DNA. So what's the following step? Cancer. Examples: papillomavirus. They cause cervical cancer. Do we know the papillomavirus? HPV, right? There is a vaccine for that, isn't it? Ladies are supposed to take it. Girls are supposed to take it. Why are you taking it? Because it's an oncovirus. Does that mean that anybody gets human papillomavirus will have a cancer? No, it doesn't mean that. It means you might, especially if it is multiple attacks, like you get it over and over, or it stay with you for some time. Epstein-Barr virus will do the same thing, but in the lymph nodes, in the lymphocytes. They cause permanent damage to the DNA. Anytime, remember this, anytime you have a permanent damage to the DNA, this is pre-cancer. This is how the cancer starts, permanent damage. So can we have non-permanent temporary damage? Yes, yes. The DNA can fix itself. If this is permanent, means it's a damage that the DNA cannot fix itself. It's permanent. It will stay broken like this. This is cancer. The third example will be, it's not here, but know it. It's good to know. Hepatitis C. They can cause liver cancer. It's another oncovirus. It's good to know. Papillomavirus, Epstein-Barr virus, and hepatitis C is also an oncovirus for the liver cells. Did you get this point? If you wanted to cultivate the virus, can you cultivate the virus in a medium that have all the nutrients that you need? No. Even if you have all the nutrients? No. No. Yes. You have to have a living cell. That's it. Even if I give you all what you need, what I need is actually nucleus. I need the DNA. This is what I need. You give me nutrients, I won't do anything because I don't even have a nucleus, I don't even have a cell membrane. I'm not even a cell, right? So you have to put it in a living cell. Okay? How can you do that? For cultivation, you isolate, you identify the virus in clinical specimen. What's a clinical specimen? A throat swab can be a specimen. You can get a cervical swab from the cervix. You take a cotton swab and get a swab whether from the throat or from the cervix. Or actually, you can get like a sample from the liver, for example. Whatever it is. And you put that injected into cells and you recognize what is this. And you can isolate. This is one. This is why we cultivate the virus. In all cases, where you cultivate living cells. You have to have whatever it is, any type of living cell. Animal, okay. Protozoa, that's fine. Bacteria, that's fine. Anything that's living, okay? Anything that's living can act as a culture, which we call it cultivation. But why are you cultivating in the first place? Number one, isolation, identification. Like, you're giving me a sample and you're telling me, can you tell me what I have? What kind of disease do I have? I'll tell you, give me a sample, I will isolate it, look at it, and I'll tell you what it is. You can make vaccines. How do they make the vaccines? Like if you got a flu vaccine, for example, right? How did you make that vaccine? Simply, they take that vaccine. This is most of the time. Not the vaccine. They take the virus from you, and they inject it into the horse. Okay, and the horse will make some antibodies. They can use that antibodies, or they can get that virus and kill it, and re-inject you. Not actual injections. You put it under the skin, or it can be attenuated virus. Attenuated means they beat up the virus until it becomes very weak, and then they inject you under the skin. In all cases, whether it is a dead virus, a weak virus, a piece of the virus, whatever it is, when they inject it into your body, it cannot infect you, but it resembles the infection as if it's not really infecting you. Okay, this is like if you take the flu virus, right? Did you take the flu virus? Anybody here take the flu virus? If you take it, you will feel as if like you're having a little bit of a flu, not really flu. Okay? So you feel some congestion and stuff. If you ask yourself, what is this? They gave you a killed flu virus. It's a virus but killed. It cannot cause a whole flu. It cannot. But when it goes into your body, your body will say, oh, okay, what is this? Is it a virus? No, it's a killed virus. Still, it's a foreign body. So I need to react, to make antibodies. So it will do some effects. This is how they prepare the vaccine. So this is one of the ways. They can also do some studies on the viral structure, the genetics, and the effects, and so on. So it can be also used to prepare treatment, for example. So what's the culture exactly? Any living cell. Can this be a tissue? Can I get some cells, some tissues? Yes, it's cells. Can I use a bird embryo? Yes, it's in the embryo of cells, so that's okay. Can I inoculate it into a living animal? Yes. Into the brain of the animal, into the blood of the animal, into the skin of the animal, it's okay. As long as it's something that's living. So here it is. This is tissue, for example. They get some tissue from animal or something, and they put the virus in it, and they see what's happening. Here is another one. They injected into the egg embryo. Whether they put it all the way in, or in the chorionic villi, or in the sac, in any part of that egg. Medical importance of the virus. Why the virus is medically important? This is the most common cause for acute infection. Like if you get 1 million person who have acute infection, most of them will be virus. Some of them will be bacteria or protozoa or helminthes or any other types of pathogens, but the big portion will be viruses. There are billions of viral infections that's happening every year. Mortality rate in some viruses can be high. Meningitis, for example, viral meningitis, and others. Chronic diseases like chronic hepatitis that can lead to liver failure. It can lead to cancer, right? Ecosystem, because the virus can invade the algae, it can invade different types of bacteria, it can invade different things to affect the ecosystem. Now, how to detect the virus and how to treat it? Of course, it's a lot difficult, Bacteria is very obvious. You look under the even light microscope, you can see it. But the virus is kind of hard. Bacteria, you can put it in anything. Virus, you cannot. So it's difficult. So that's why doctors actually, doctors or nurses or PA or something, when they do the diagnosis, they do not count on how the virus looks like and all that stuff. They count more on a clinical picture. Okay? Like if you're coming to me with the flu, I don't have to get a sample and do all of that. That's too much. Okay? Too difficult, too much, costly, time consuming. So they count more on a clinical picture. Okay? You're coming to me, I'm coughing and sneezing and I have congestion, I have runny nose, I have fever. Okay, so you have flu. It goes like this. If it is something that's taking longer time, okay, this is where I can do more research. Is that the case? If I need to do more research of this, or this is something that I cannot know for sure what it is. It can be something that's more complicated or something that's more dangerous. Okay? If that's the case, you have to give me a culture. And in that culture, what I'm looking at, I look at the virus itself, and I look at, most importantly, this, cytopathic effects. Okay? You look at the Negri bodies. Which virus gives you the Negri bodies? Rabies. In which organ? Neurons. In the neurons in the brain and the nervous system. Yes. Okay? How about downy cells? Mononucleosis. Mononucleosis. Affecting which cells? What are these downy cells? It's a lymphocyte. Downy cells are lymphocytes, swollen lymphocytes. Okay? So these are some of the cytopathic effects. If I see it, I tell you what it is. Okay? I can study the virus itself. And also, another way that's very important, show me the antibodies and I'll tell you what it is. Right? Like if you give me a blood sample, can you tell me if I have hepatitis or not, hepatitis or not? Give me a blood sample. What are you doing with the blood sample? I'm doing two different things at the same time. I'm looking at the virus, okay? And the cytopathic effects. This is one. The other thing is, I'm looking at the antibodies. If I find antibodies against viral B, you have viral B. That's it. If you have antibodies against virus C, you are infected with virus C, right? So I can detect the antibodies, which is the immune system. Okay? The question is, why don't you just give me antiviral drugs? Well, the antivirus are, most of the time, we don't even use it. This is a last resort. Usually the virus will go by itself. Okay? We can give you some supplements, give you some antibodies, give you something to augment your immune system. But to go to the antiviral medications, the problem is antiviral medications, most of them have a lot of adverse effects. They do bad effects. So unless I really, really need it, I'm not going to use it. Okay? Can you give antibiotics to the virus? Of course not. Antibiotics are for bacteria, right? Why? Because you know how the antibodies work? They either work on the membrane, on the cytoplasm, or on the nucleus. Does the virus have any of those?
No.
So it doesn't work, right? How does the antivirus work? Simply, any of the steps that we talked about. This is how the antivirus work. Preventing adsorption? Yes. Attachment. Preventing penetration? Yes. Preventing synthesis and replication? Yes. Preventing assembly? Yes. Any one of those. This is simply the virus, but unfortunately it have a lot of side effects. Prions, this is what we talked about before. It's an infectious defective protein. What is it again? Infectious defective proteins. Infecting proteins? Yes. It's not even living. It's not something living. It's a protein that if it gets into the cells, it will cause infection. And the problem is sterilization doesn't remove it. Most of the sterilization doesn't remove it, unlike other things. And we talked about those before, spongiform encephalopathies. What are the two examples of the spongiform encephalopathies? Can you remember one of them at least? Mad cow.
And Jacob Cross.
Yes. Creutzfeldt-Jakob disease. Creutzfeldt-Jakob disease. These are spongiform encephalopathies. Are these diseases caused by virus or bacteria or protozoa? No, it's none of those. Is the prime as a virus? It's not even a virus. It's not a virus. What is it? Defective infectious protein. Defective infectious protein. Misfolded protein. Just a protein, no genetic material? Yes, just a protein. So here it is. Spongiform encephalopathies like Creutzfeldt-Jakob syndrome, mad cow, also known as bovine spongiform encephalopathy. So spongiform encephalopathy can be in humans and can be in bovines, which is cow. In humans, it causes Creutzfeldt-Jakob disease. In cows, it's called it caused the mad cow disease. Others are like scrapie in sheep and goats, wasting disease in elk, and so on. These are different types of prion infection. Other infectious agents. What do you mean infectious agents? Like, is it a virus? It's a kind of virus, but different. That's why they call it agent. Why don't you just call it virus? It's not really a typical virus. It's not. Is it a bacteria, protozoa? No, it's none of those. So what is it? We call it agent. We call it an agent because you can't really put it under one. It's similar to the virus anyway, but it's not a real typical virus. Satellite virus is one of them. The satellite virus depend on other viruses for replication. Is that typical virus? Do you have to wait for other viruses to invade and then you follow them? Right? So the normal virus is I go into the cell, attach, invade, replicate, right? This doesn't do that. If you go to your cells, it will not do anything. What is it going to do? It will wait for other virus to come and invade, and then I will jump on you. Right? Do you get the idea? I need another virus. A virus that needs another virus. Satellite virus. Viroid, what's oid means at the end of any word? Like or similar to. Viroid is something that's similar to virus. It does not have a protein coat. Viroid basically invade plants, mainly. Okay, can you tell me the answer? What do you think? C, prions? Yes, does prions have any genetic material? No. But animals have genetic material? Yes. Bacteriophage, satellite virus, viroid, they all have genetic material. Okay, so it is. This is a microbial deck. This is not really needed for this exam, for this chapter, but this is just giving you examples that you can use for the microbial decks or microbial flashcards. Did you start doing that? Okay, that's great. So it can be used, but it's you're not going to be asked about it like in part of the next exam. And that's it for this chapter.